Hello everyone, welcome to the class of biochemistry, and in this video I'm gonna show you some of the instrument that is used in the biochemistry laboratory and their handling. Before beginning with the topic, let us begin with the objectives. The objectives deals with the study the handling of electronic balance, water bath shaker, PS meter, flame photometer, hot air oven, centrifuge, magnetic stirrer with hot plate, spectrometer, SemiAuto Analyzer, and Calorimeter. So now let's start with the very first instrument, that is electronic balance. It is a device which is used for the accurate measurement of the weight. Talking about the principle of electronic balance, the accurate measure of weight is achieved using the device called load cell, which act as a transducer. The load cell is a Wheatstone bridge wire to give a small but accurate voltage, usually in millivolts, to stable electronic amplifier. Now let us talk about another instrument that is used in biochemistry laboratory, and that is water bath with shaker. It is a compact water bath with shaking capabilities. A digital thermostat is fitted for the case of use. It has a wide range of shaking speeds and temperature digitally displayed with a high temperature controlled accuracy. Talking about the principle of water bath with shaker, the water bath with shaker is used to carry out various chemical reactions at a specific temperature with constant shaking to carry out the reaction faster depending upon the requirement of the experiment. The temperature of the water bath is controlled by a thermostat arrangement. Let us now focus on the uses of water bath with a shaker. It is used for the experiments which requires different temperature for determination of chemicals like serum analysis, serum enzymes, urea, cholesterol, glucose, triglycerides, and so on. Now let us start another important topic, important instrument, that is pH meter. A pH meter is an electronic device measuring the pH, acidity or alkalinity of a liquid. It consists of a special measuring probe, a glass electrode connected to an electronic meter that reads and measures along with displays the pH reading. Talking about the principle of pH meter, the probe measures pH as the activity of hydrogen ions surrounding a thin wall glass bulb at the tip. The probe produces a small voltage, about 0.06 volts per pH unit, that is measured and displayed as pH unit by a mirror. At neutral, the voltage at the probe is equals to 0 voltage. Talking about the uses of pH meter, it is used to determine the pH of a solution. Let us emphasize on another important instrument as flame photometer. It is a device used to determine the concentration of certain metal ions. The sample is introduced to the flame at the constant rate. Talking about the principle, it is a controlled flame test with the intensity of flame color qualified by a photoelectric circuit. The sample is introduced at constant rate. Talking about the uses, it is used in laboratory to find the concentration of certain metal ions. Let us briefly explain about hot air oven. It is an electronic device used for sterilization. It uses hot, dry air to sterilize the particles. About the principle of hot air oven, when electricity is passed through the heating coil, the electrical energy is converted into heat energy, and thus the temperature is controlled by the thermostat and dry air. Talking about the uses of hot air oven is mainly used for the following purposes: to dry heat sterilization of glassware, heat stabilized substance, along with the preparation of anti-coagulated bulbs and drying of glasswares. Let us now talk about centrifuge. It is a device which is used for the purpose of separation. Let's talk about the principle of centrifugation. The working mechanism of centrifuge machine is based on the principle of centrifugal force, and that is, it acts via substances in a circular motion. Uses of centrifuge machine is to separate the serum of plasma from the blood cells, sediment the urine, along with washing of RBC by a normal saline. Another important topic that we deal is about the magnetic stirrer with the hot plate. So what is magnetic stirrer with a hot plate? It's a device which consists of a rotating magnetic field to cause a stirrer immerse in a liquid and to spin it very quickly, thus stirring it. Talking about the principle, in a magnetic stirrer, the rotating field may be created either by rotating magnet or a set of stationary electromagnets placed beneath the vessel with the liquid. Magnetic stirrer often include a hot plate or some of other means of heating of a liquid. Talking about the uses of magnetic stirrer with a hot plate, the instrument is used for fast mixing of the solutes in the solution, and the hot plate helps to provide the necessary heat. Now let us focus on another important instrument that is used in biochemistry laboratory, which is none other than UV spectrometer. It's a device for the quantitative measurement of the reflection or transmission properties of a material as a function of wavelength. It deals with the visual light, near ultraviolet and near infrared, but does not cover the time resolved spectroscopic techniques. Talking about the principle of UV spectrometer is based on Beer's and Lambert law, which states that optical density is directly proportional to the concentration of the colored solution. Now let's focus on the uses. Talking about the uses, it is used to measure the intensity of the light. Its important features are spectral bandwidth and linear ranges of observations or reflectance measurement. Talking about further uses, it helps in the measurement of transmittance or reflectance of a solution. Now we talk about another important topic, that is SemiAuto Analyzer. So it is a device which is used in the laboratory for analyzing the chemical solution. Now let's talk about the principle of auto analyzer. The analyzer are called semi-auto analyzer because the initial stage of the specimen analysis, likely like pipetting, reading endpoints, similarly displaying the test result, printing and memorizing these results, and graphs are done by the technique. However, its uses are like it is used in immunoassays, testing coagulations and drug assays. It helps to plot the absorbance versus wavelength for selected ranges. One of the most important devices or instrument used in biochemistry lab is colorimeter. It's a device used in colorimetry. It measures the absorbance of the particular wavelength of light by a specific solution. So it's the principle of colorimeter is it suggests that the concentration of the solute is proportional to the absorption. Talking about the use of colorimeter, it is used to determine the concentration of solute in a given solution by the applications of Biot-Lambert law. So thank you so much guys for watching my video. I guess it was very fruitful video to you. Keep tuning on. Cheers.